um, plans for this project and what's been done so far, and what he's been doing to lead up to this work.
Yeah, so thanks, Jackie, for the introduction and for inviting me here and giving me the chance to present some of our work. So as mentioned, I work in the Brigham and Women's Hospital at the newly formed Jean Lay Institute. So today I'm going to talk about a collaborative work with the Wang Lab over at MGH, where we've studied pancreatic cancer, and in particular trying to understand sort of mechanisms of therapeutic resistance. So pancreatic cancer is a relatively rare type of cancer, but unfortunately, it is associated with a relatively high mortality rate, as you can sort of see here. So this is numbers from the US, where you can sort of see that both for males and females, it ranks as one of the deadliest cancers that we have. So there are a couple of main reasons for why it is this way. So one reason, which I'm not going to talk about today, is that pancreatic cancer is often detected at a relatively late stage, which of course makes it harder to treat. The other reason is that pancreatic cancer, and in this talk I'm going to particularly be talking about is the most common and also most lethal subtype, which is called PDAC, or pancreatic ductal adenocarcinoma. It has one of the really hallmark features is that they tend to develop resistance to the chemotherapy and radiotherapy that are typically used. So one of the problems that the field is really struggling with is sort of how you can avoid this therapeutic resistance from emerging. So the main background work for the talk today is another paper that came out from the Wang Lab last year. So in this project, they used an older spatial transcriptomics technology called geomics to profile tumor biopsies. And the main drawback of the geomics is that it has sort of lower spatial resolution. But despite that, they were able to sort of dissect these different tumors sort of molecularly. And the big take-home message from this paper is that in addition to the sort of two previously known types of subtypes of malignant cells, which are typically referred to as the basal and the classical, they found a third one, which they refer to as NERP, or neural-like progenitors. And the bad news is that these NERP cells are associated with the worst clinical outcome. You have the worst type of prognosis if you have a lot of these in your tumor. Whereas also the basal and the classical also have different associations with the clinical outcome. So we wanted to sort of build on this study and sort of better understand what are the sort of specific types of interactions that are happening with these different subtypes of malignant cells. So to do this, here is the sort of the study design that we set up. So we used a newer spatial transcriptomics technology called the CosMx platform from NanoString. And we have a total of 15 specimens, 15 samples from 13 different donors. We have sort of two different types of panels that we're using to probe these. They're called the standard and the custom. I'll tell you more about them later. And then the samples are either untreated or treated. So this is before treatment or after treatment. So this allows us to sort of infer what are the changes that are happening in response to treatment. So the main caveat here, and the main limitation of this study, is that the samples are not matched. So the untreated and the U1 and the T1 are not from the same donor. So that obviously limits the amount of causality that we can infer from the findings here. And then, so after this sort of experimental treatment, we did various types of computational analysis, which is sort of what I'll be talking about today for the most part. So a little bit more about the method. So this CosMx instrument is based on something called the spatial molecular imaging, or SMI, where the idea is basically that you have these sort of fluorescent dyes that get attached to probes that hybridize with RNA transcripts. And then you do this sort of in multiple rounds with 4 different colors. And then you sort of get a readout in terms of a code, and based on doing that over multiple rounds, you can then infer sort of which transcript it corresponded to in a certain point. So the problem with this technology, unlike sequencing, is that you need to define your probe set in advance, so it's not unbiased. So in the standard setup, we have 960 sort of pre-selected probes. The good news is that these probes were this panel was specifically designed towards cancer. So pretty much all of the probes are useful to us. And in the custom, we're allowed to choose 30 probes ourselves. And those probes were chosen to specifically be able to distinguish and identify these 3 different malignant subtypes that I mentioned before. So here is just an overview of what it looks like. So on the left here, we have the standard H&E staining from the biopsy. And then you need to choose on the order of between 20 and 30 fields of use, or FOVs, that are sort of shown here as rectangles, which are the ones that get actually profiled, and that are ones that we'll be looking at for the rest of this talk. So here's one example. We've sort of colored each of the transcripts that are identified sort of based on these 3 broad categories. So the green ones, so these are genes that are sort of associated with the malignant states. The blue ones that are associated with the fibroblasts, or the cancer-associated fibroblast CAFs. And finally, the magenta ones, which are sort of immune type of genes. And most of the immune genes, our immune cells here are macrophages. And the type of characteristic pattern that you see here is that you have these sort of gland-like structures here formed by the malignant cells, and then they're interspersed by these CAFs and the immune cells. So the first thing we needed to do was to sort of do this sort of annotation in a little bit more fine-grained manner. And for that, we set up the workflow, which is sort of shown here. So first, we use a sort of pan-CK marker, which looks at multiple different types of keratins to split between malignant and non-malignant cells, which you can sort of see works quite well here in the UMAP. And then for the UMAP, for the non-malignant compartment, we use a sort of supervised clustering approach, which requires single-cell or single-nuclear RNA-seq data from a sort of matched sample in order to be able to do these assignments. As I mentioned, we also have multiple different donors. We need to correct for that. As you might know, there are quite a few different methods around in single-cell RNA-seq world for doing this. Most of those are not applicable since they operate in a latent space, but we need the actual gene expression matrix in order to be able to use the supervised clustering approach. And so for that, we used an old school method called LIMMA. Some of you who've been working since the time of microarrays might be familiar with it. And it seems to do a good job in terms of sort of mixing the batches, as you can sort of see here in the UMAP. Then we went about and we were able to sort of assign these different cell types here. And as you can see here from the markers, they're sort of they're isolating the different types of clusters quite nicely. We're also able to sort of go in more deeply the malignant cells and split them up into these three NERP, basal, and classical substates. And for the CAFs, we can identify the MyCAF and the ICAF states that have been described previously in the literature. So here is again showing you the UMAP. And you can sort of see here that the different clusters or cell types separate quite nicely. One difference here is compared to what you might expect from a single cell RNA-seq experiment is that the different clusters are not quite as crisp. And we think that this is mainly due to the fact that we have only have these 960 probes, and hence it doesn't allow us to sort of separate them as nicely as you would have with a single cell RNA-seq data set. So the overall sort of trend that we see between the treated and untreated samples is sort of illustrated here. And the big difference really is a big loss of malignant cells between the two conditions that you can sort of see here. Whereas with the non-malignant cells, there's a small drop, but it's not statistically significant. And then again, sort of looking in the malignant, non-malignant subcompartment, you can sort of see that we have some shifts in terms of the proportions of cells between the untreated and the treated. So one advantage, of course, with the spatial transcriptomics data is that we can sort of map everything back to all these annotated cells back to their original location, and then we can sort of start delving into it further. And this is sort of shown here. Again, we're just looking at the densities across the different types of the different samples and the different treatment categories, and you can sort of just by looking at this at a high level, you can sort of appreciate that there are some patterns here, and it does turn out that if you sort of try to train a classifier, you can sort of using the different cell densities here, you can do a good job at separating the treated versus the untreated the cells. We can also look at the different malignant substates, and again here you can sort of see as consistent with the previous paper where they had anticipated that the NERP are sort of between 10 to 15% of the total number of malignant cells. You sort of see here, and then you have a sort of mixture of the classical and the basal states for the remaining malignant cells. So now we wanted to sort of better understand the role of these different malignant subtypes. So the first question we asked was really sort of what kind of sort of heterogeneity do we have in these? So as I mentioned before, the characteristic feature of these FOVs is really the presence of these sort of glands. So what we did was that we devised an algorithm, which is sort of based on the classical DB-scan algorithm, to extract these different types of these glands, and then we looked at each gland individually, and we asked sort of what kind of mixture between the three different substates do we have. And you can sort of see the overall the global picture here, and I think the overall message really is that for the most part the glands sort of tend to be dominated by one of the three types, and there's a really only a minority of the glands where you have sort of reaching somewhat equal proportions of the different subtypes. And we think this sort of makes sense because our hypothesis is that these glands would be sort of starting from a single clone or a single cell, and this sort of implies that the switching between these different malignant substates is a relatively rare event. So we can try to sort of quantify this heterogeneity, and for that we use sort of two different indices. So we have a heterogeneity index, which is sort of illustrated illustrated schematically here. And then we have this interspersion index, which really just shows, tries to measure sort of how often the different cell types sort of sit in between one another. And again, sort of looking at the data and comparing to a null model where we assume that everything is sort of all the subtypes are sort of randomly assigned. What we sort of see here is that we have the expected heterogeneity is much higher than the observed heterogeneity here, comparing the fill lines, whereas the interspersion is actually sort of pretty well tracking the expected levels. So again, this is a sort of confirmation of what we had here at the bottom, that you do seem to have these sort of blocks to a much larger extent than you would expect. So the next thing we wanted to investigate is sort of really to understand how these different sort of malignant glands, how they interact with other cell types. And for that, it's sort of something that has been looked at quite a lot, in particular in the sort of dissociated single-cell RNA-seq literature. And particularly what people have focused on is sort of ligand-receptor interactions. So then the idea here is really to sort of find sort of cell types or pairs of cell types where one of the pairs has sort of upregulate or high expression of a ligand, and then the other cell type has a high expression of the matching receptor. And sort of based on that, you'll assume that these two cell types are sort of probably communicating using this pathway. And then you're sort of looking for pairs of cell types that have sort of lots of ligand receptor pairs in common, and you're saying that these two guys are probably talking to each other quite a lot. So intuitively, you would expect that if you have spatial transcriptomics data, you should be able to somehow leverage that and do better. And when we started this work, we couldn't really find any methods that were capable of doing that, because up until so far there hadn't really been this sort of high resolution spatial transcriptomics data hadn't been available. So the methods were unable to take full advantage of the resolution that the CosMx panel provides. So the overall idea of the method is sort of illustrated schematically here. So what we do is that we focusing on sort of a pair of cell types, so a source and a target, and we first do a sort of use a DB-scan approach to sort of identify clusters, and then we say that clusters that are located far apart from another, they are sort of unlikely to be communicating, and hence we're going to exclude those from further analysis, because they don't have the sort of the matching pair to talk to each other. So we're only going to be use the ones shown here in the full circles, where we have a sort of where we think that there's a good chance that the ligands will be able to reach their target receptors. So the next step is sort of for each one of these sort of where we have these close pairs that are close in space, we use an optimal transport algorithm to really to figure out sort of to sort of solve the problem essentially of how to best move the ligands to the receptor. So again, optimal transport is a sort of mathematical framework, which again, conceptually, you can think of it as you have a sort of a pile of dirt over here, and then you have a bunch of holes over here, and now you need to figure out the optimal way of moving the dirt pile into the holes. And again, really from a computational point of view or mathematical point of view, the key thing that we need to do here is to sort of devise a cost function that somehow balances the differences in the ligand and the expression levels and the spatial distances. And from first principles, it's not really possible to do that because we don't really know what those are. So what we ended up doing was sort of coming up with sort of what we think were sort of some reasonable suggestions for cost functions that combine the spatial distance and the ligand and receptor expressions, and then sort of make sure that the results are robust with respect to the choice of cost function. So once we've done that, we can sort of calculate a likelihood that any given pair of cells will be communicating, and then we can sort of aggregate those likelihoods, and then we can do sort of controls, which turns out to be the best ones are permutation tests in order to sort of establish a baseline. And then we can sort of calculate sort of the total likelihood across all of the different sort of pairs here that these two cell types are communicating, and then we sort of can compare these likelihoods between the treated and the untreated conditions, and sort of find out which ones are likely to be have changed between with regards to the treatment. So here is a sort of is a high-level summary of these findings. So what you can sort of see here are the different cell types shown here as dots, and then the arrows sort of start from the source producing the ligands and point to the target where the receptors are. And so if you look at these two results for the untreated and the treated, it's not hard to appreciate that the biggest change that we have is from the calves to the malignant cells. So this is what we're going to start focusing on, since that was the biggest change. And this is, of course, also something that has been reported in the literature to be an important interaction. So it was by no means surprising. So here is a good old-fashioned volcano plot to sort of illustrate this result. So here on the x-axis, we can sort of see the fold change between the sort of treated. So here's things that are higher in the treated versus the untreated conditions, and then the p-values. And then we can, so each dot here will represent sort of one ligand receptor pair that is involved in the communication between the calves and the malignant cells. So here is another a waterfall plot, which is tries to represent the same type of data. The difference here is that we've sort of considered three different types of p-value tests, because again we don't really know what is the appropriate model, statistical framework to use here. So we use three different ones, and we sort of see that they seem to be in reasonably good agreement, and we're picking out sort of several things that appear to be significant by more than one measure. Another nice thing that we can do with this is to again map it back to the original spatial context and visualize the results. So here I'm showing you results for sort of two of the best candidates that were picked up. So at the top level here, or top row, we have the untreated, and then we have the treated. And what we're showing is the ligand in yellow and the receptor in blue. Each dot represents a cell. The size of the dot corresponds to the expression level. And then you can sort of see that some of the dots are faded or more transparent. So those would represent cells that didn't have a sort of a partner, but from the first step where we sort of select the ones where the clusters are nearby. Whereas the ones that are in sort of deeper colors have a partner nearby. And here you can sort of visually, you can very easily appreciate that there is a big difference between the treated and the untreated, in the sense that here at the bottom, we have sort of many large dots that are sort of sitting close to each other of different colors, whereas here the dots are much smaller, and they're also sort of typically sitting further apart. So this, I think, is a sort of, we can appreciate that the algorithm has indeed sort of picked up a big change between the two conditions. So we can do sort of several additional controls in order to sort of confirm these results. So I'll start with a few computational approaches. So the first one is again quite straightforward, and it's a nice one because it's also sort of generally applicable. It will work for pretty much any data set, because as I mentioned before, in order to do the annotation, we need to have matched a single nuclei or single cell RNA-seq data. So we can leverage this data again, and we can sort of compare the same sort of the differences in ligand and receptor expression from the two conditions in this companion single nuclear RNA-seq data. And as you can sort of see here, is that we have a sort of weak but significant correlation, and sort of several of the top candidates are still coming out in this other data set. So this is a good indication that our algorithm is picking up something useful. Another control, which is again nice because it's sort of quite universally applicable, is that we can sort of go to sort of various sort of pathway resources such as KEGG, and then we can look there and we can sort of see at what's annotated as sitting sort of downstream of the receptors. And then we can look in our data, and depending on if the receptor is predicted to be activating or repressing the things that are downstream, we can then look into our data and see if this is confirmed. And this is again another example where it comes out quite nicely. We do see that there is a sort of weak but yet significant correlation, particularly for the activated, to a somewhat lesser degree for the repressed genes. I think the main reason why you have problems with the repressed genes is because we have a sort of the dynamic range is relatively low, and just like single cell RNA-seq data, the cosmics data is quite sparse with a lot of zeros. So hence, sort of detecting sort of going down from an already very low baseline is very difficult. So that's why I think that why this doesn't seem to be working as well here. We can also ask, or one possibility is that these changes that we're seeing is just happening sort of across the board, and it's not really related to these cells being neighboring. So the way that we try to address this question is by splitting the cells into these two different categories. So the interacting cells are the ones that are sort of near other cells in the other category, whereas the non-interacting cells are the ones that are sort of further away and from their partners, and hence unlikely or algorithm decided that they were not communicating. And as you can sort of see here, is that we do see a difference in sort of 3 of the 4 cases here between the interacting and the non-interacting cells for both the ligand and the receptor. So again, this analysis really implies that what we're seeing is sort of important for the interactions, and it's not some sort of epiphenomenon of a sort of global change in expression. So finally, we also did a validation experiment, and by we, I of course mean the Wang lab. And what they did was that they did a sort of organoid experiment where they do these sort of separately culture the tumoroid organoids and the calves, and then they get inserted into a mouse. And then they use this 5-FU treatment to simulate the chemotherapy. And then we do single nucleus RNA sequencing afterwards, and we can compare the 2 different conditions. So here is the sort of the UMAP from the RNA single nuclear RNA-seq data. And again, we can crunch the numbers, and we can compare the expression levels of the ligands and the receptors. And again, correlating with what we had from the Cosmics SMI data, we again observe a weak but significant correlation, again confirming that we are picking up a meaningful signal with our algorithm. We looked a little bit more closely, in close detail, at the sort of top hits here. We noticed that many of them were from the IL-6 family. And this is again not super surprising, because it has been shown in the literature, and we try to dissect this a little bit further, sort of by breaking it down, both in terms of the different types of calves, and we sort of see that we have more of the IL-6 ligands from the I calves compared to the my calves. And then when we look at the distances from the different malignant subtypes to the ICAFs, we see that the NERPs tend to be closer, followed by the basal and the classical. So why is IL-6 problematic? It is associated, it activates the JAK-STAT pathway, and it's IL-6 is a bit complicated, but on the receptor side, there are sort of two types of receptors, the IL-6-ST and the IL-6R. And what people have shown is that it's a relative ratio between IL-6-ST and IL-6R that determines the amount of JAK-STAT activation. And this is replicated in our data. We sort of see that there is a good correlation between the two. We can break this down by the malignant subtypes, and then again you see here that you have the highest IL-6-ST to IL-6R ratio in the NERP, which would again then be predicted to have the highest JAK-STAT pathway expression, which is again consistent with a poorer outcome associated with the NERP's subtype. Finally, we also did an experiment where we added IL-6 to 3 different pancreatic cancer cell lines while they were subjected to a 5-FU treatment. And we see here that for 2 of them, we have a sort of significantly increased degree of survival for these malignant cells. So this also shows that the IL-6 helps survivability for the malignant cell, which is consistent with what we had found. So next we decided to look at further interactions. So the second biggest change in interaction is from the malignant to the macrophages, as you can sort of appreciate here. So we basically run the same pipeline as before, and here you can sort of see the volcano plot. You have numerous interactions that are enriched in the treated and a smaller number in the untreated cells. We looked at some of the top ones here, and for example, the CSF1R, there is support in the literature to sort of show that if you inhibit this, then this in the macrophages then results in enhanced T cell response to the cancer. So this is consistent. The same was found on the IL-34 side, where it was shown that if you block this, that it improves the response to immunotherapy and PD-1 treatment in particular. We did a similar thing with a sort of third most important, or third biggest change, which is the calf to macrophage signaling. And here again is a volcano plot, and this top here between GAS6 and MERTK is also there's also support in the literature that this results in more immunosuppressive and tumor microenvironments. So overall, we're quite happy with these top hits. We can do the sort of similar types of analysis that we did before with the and looking at the downstream targets. And again, you can sort of see here that in this case, the directions are sort of what we would expect, even though we don't reach statistical significance, but that we do that here in the calf to macrophage interaction. So with that, I wanted to sort of change gears a little bit and talk about a second arm of this project. So one of the problems with the spatial transcriptomics assay is that it is relatively poor at distinguishing different immune states and cell states, so for that we can use a different approach, which is to use a codex technology, which is also spatial, and it profiles protein cell surface markers. So this is by the a company called Akoya. They have it's a 103-plex platform, which is sort of targeted specifically at immune cells, and we can do this not at exactly the same samples as we did with the Cosmics, but we can do that at consecutive slides. So it gives us a very close approximation, and it gives us a better way of looking at or annotating the immune cells in order to help understand how they relate to the malignant cells. So here's an example of what this data looks like. So here's sort of the H&E slide with the FOVs selected for Cosmics. The Akoya is nice because it actually gives us the whole tissue and not just the FOVs, but for the purpose of this, we'll mainly be focusing on the FOVs, where we have overlapping information from the Cosmics panels. And here in green, I've sort of highlighted what we call as malignant cells, so you can get a chance to understand what that looks like. We do cell type annotation. Since the data modality is different, we use a different approach, but again, it's relying on a published method, which is basically uses a neural network which has been trained on other data to take the protein surface markers and assign them into these different cell types. So it's obviously helpful to compare to the SMI data, and what we can sort of see here is that the big difference is really in the calves, which are sort of at a relatively high proportion in the SMI, but almost pretty much absent in the Akoya, which is not surprising because they have very few markers that correspond to them. But if you focus on everything else, you can sort of see here that the highly abundant cell types, which are sort of marked here in green, they have relatively good correlation in terms of their abundance compared between the two platforms. And I should also mention that what's not shown here in this bar plot is that there was a sort of high number of unassigned cells, which that number pretty much matches the numbers of sort of presumably missing calves. So this makes a lot of sense. And what I should also say is that we have here, this is a pilot project, so we only have 4 of the donors that we had for the Cosmics panel that we're profiling. So now we can sort of start looking at this in more detail. And I think sort of one of the main take-home messages that we found here is that there seems to be a lot of variability in terms of the immune landscape. So here you can sort of see zoom in, and what we can appreciate is that we have sort of regions that are not too far from each other, where you have sort of either sort of high levels of T-cell infiltration or sort of more moderate levels. And here, where there is sort of hardly any T cells, but lots of macrophages. So there's clearly a lot of heterogeneity in here. And what really sort of surprised us was when we sort of looked at this more globally, was that we saw these large numbers of sort of lymphocyte aggregates, so mixtures of T cells and B cells. And the reason why this is surprising is because traditionally, PDAC has been considered as a sort of very sort of to have a very immunosuppressive environment and to have a very sort of low presence of immune cells. But this sort of suggests that conventional view may not be sort of super accurate. So this is sort of work in progress, but we're sort of really trying to understand sort of what is going on here. And given that these are sort of mixture of T cells and B cells, it's sort of it's promising to try to call at least some of them as tertiary lymphoid structures or TLSs. But we really do think that there is there's a whole span here of different types of maturity. So one of the things that we did was to look at various ligands that have been associated with or been labeled as TLS organizing factors in the literature, and we can see sort of which cell types seem to be producing them. And here we can sort of see that there does appear to be some cell types that really stand out as being sort of more responsible for these types of molecules. And I think what's particularly interesting here is the CXCL12, which seems to be produced by the ICAFs. So we can also now try to sort of use this information in order to try to understand the interaction between these different immune cell sort of states and aggregates and the different malignant glands, which, as you might recall from before, tend to be sort of dominated by one of the malignant subtypes. So this is because of the this is conceptually it's straightforward, but technically it's a little bit tricky because of these consecutive slides. So again, these differ by about 5 microns. So it's unlikely that we're going to see the same cell in the two consecutive slides. And so here is an example of sort of what it looks like. And you can sort of, if you look at it by eye, you can sort of reasonably sort of match up the different glands that we're seeing here, but it becomes a little, when you get into the deep details, it becomes a little bit more complicated. So we devised a sort of computational workflow to do this, and the basic idea here is to sort of find some really well matching glands, and then we use those as anchors. And then we can sort of shift around the other things, and we can do the sort of final alignment. And we can, based on that, we can sort of identify which immune cells are within a certain distance of a gland, and we can start doing comparisons. And this is sort of one comparison here. And now you can really start to see here that there are this hypothesis that we had, that there are these different glands are associated with different immune environments, seems to be holding up as they are indeed sort of surrounded by different types of immune cells. So this is sort of very much ongoing work. So I don't really have any firm conclusions around this, but we're hoping to be able to wrap this up pretty soon. So in summary, I've told you today about how we used spatial transcriptomics to characterize the tumor microenvironments of PDAC. And this allowed us to sort of identify all the major cell types and in space, and sort of characterize their sort of the relative locations. In particular, we focused on the malignant cells and asked how those different subtypes were found or were located. We developed a new algorithm called SCOTIA for incorporating spatial information when inferring the ligand and receptor interactions. We focused primarily on the CAF malignant interaction, since that was the one that was most affected by the treatment. And through these various validation experiments, we sort of conclude that the algorithm is outputting some reasonable things. And finally, I showed you how we started to use spatial proteomics to further characterize immune states. And with that, I'd like to mention that this method that we developed for the ligand receptor interaction is called SCOTIA. It's currently available through GitHub. It's primarily a Python package, but there is also an R version, or it will be coming soon. And if you wanted to try it, we'd be very happy to have any feedback since we're actively working on it. And finally, I would like to thank the people who did the all the actual work. So all of the computational work presented here was really driven by Jingi, who's a postdoc in my lab. And it was in close collaboration with the Wang lab, in particular, Karina, and also people from NanoString. And Ryan Park helped out with the proteomics analysis. So with that, I'll thank you for your attention. I'll be happy to take any questions.
Thanks, Martin. I was curious, it's a very tough area, right? Because you care, in some sense, it seems you care a lot about the cell surface biomarkers, right? But their sort of standard behavior isn't what happens in the tumor cells, right? So the things they inhibit may not be the same. The things that they upregulate may not be the same. And I'm just curious, what kind of an assay do you think would help? If you could sort of invent anything, what would you want to measure to try to get a better sense of the sort of receptor ligand behavior?
Yeah, I mean, I think really the first thing that I really want to be able to do, right, is to sort of spatially profile I guess the receptors is already feasible, but really spatially profile the ligands, right? Because what we're doing, right, is that there is a big assumption, assuming that the ligands, the RNAs that we're observing, right, are translated and then excreted and taken up. So to me, that's the biggest the biggest set of assumptions that goes into this algorithm here.
Right. Thank you.
Ludwig?
Yeah. Yeah, Martin, thanks for a great talk. I have a couple of questions. I don't know whether we have enough time for that, but one question that I was wondering, I'm not super familiar with this Cosmics platform, but one thing that I thought it can do, it actually can spatially profile transcripts and proteins together, right? So it claims at least to be able to do like 1,000-plex mRNAs and 65-plex proteins. I was wondering why it was necessary to kind of like do Codex and Cosmics separately.
Oh yeah, that's a great question. So I think that wasn't available at the time when we were doing these experiments. So this is sort of data analysis of data that was generated more than a year ago, I think. So that's the simple explanation. So the short answer is that you're absolutely right, and I think that sort of in the very near future we're going to sort of have these sort of proteomics and transcriptomics from the same slide, and that will obviously make life a lot easier. So
right, yeah, I think so. The other question that I had were around these three subtypes, in particular this new subtype. So what people focused on TCG bulk tumors, at least, that a lot of these subtypes are to a certain extent, like sampling artifacts, right? So different like cell type admixtures, some are where you sample more immune cells or you sample more stromal cells. So I was wondering for the third subtype, whether there's any evidence in the literature that this is indeed like genomically different, so that it has like certain, like different somatic mutations in
Yeah.
red cells, whether there's anything known in regard to
Yeah,
that?
I think it's a great question. I think the answer is probably no, that I don't think they have that information yet. I think what they may have done would be sort of basically inferring the copy sort of mutations and copy number variations and so on from the RNA-seq data, which I haven't seen, but I'm not familiar with. Again, this was only published sort of a little over a year ago, so I don't think I think they have sort of functional experiments to sort of suggest that they are sort of have different phenotypes, but again, I think what you're suggesting is obviously would be helpful.
Any more questions, Tyrone?
Video for whatever reason. I'm interested in thinking, how do you plan on integrating this with other modalities such as proteomics, metabolomics? I know that this is Perfin embedded slide right there. I don't know if you can do any additional spot checking for any like epigenomic changes or maybe methylation states, for instance, because this seems very, very constrained to the microscope slides right there. I'm trying to figure out if there's a good way, if you have any ideas on how to go across different modalities, either with these samples or with the data that we have from Cosmix.
Yeah, I think it's a good question. I honestly haven't thought much about that. I think I sort of feel that we still, from a computational point of view, there are still sort of tools to be developed for sort of extracting more out of the modality and the data that we have already. Obviously, if you had more, you could probably do something with it, right? But I think there is still some sort of not too high hanging fruit to be gotten out of sort of what we have.
Is that due to the technique from nanostring, or is that due to whatever? Is that from the data pipeline? Because I know Cosmix has some weird FOV stitching for data duplication. I'm wondering if that's why this technique was low. It's hard to be more generalized, or if that's
actually I don't know the answer to that. It's actually not. I don't know enough about the underlying technologies to say something really confidently about that.
I see. Thank you.
Any more question for Martin? Well, if not, thank you for a great talk, Martin. Next week, we have Dr. Beth Chimini from Broad Institute joining us for a hybrid seminar, and we hope to see you all then. Thank you, everyone. Have a great day.
Thank you very much.
Thanks.